I'd like to thank Vishal and the organizers for inviting me here. I was coming to Delhi for the Photonics 2008 meeting, and I was invited to come and talk at this. And my initial thoughts were, what do I have to contribute to this talk? I don't do any work in nanosystems, and I'm primarily a theorist and a biophysicist at that. But then thinking more about it, I decided, well, we are talking about quantum computing systems, and what is the one computer that all of us have that outperforms any silicon-based computer, brain? And decided I got into doing work on human vision primarily because I was interested in how single photons were converted into electrical signals. And the fact that we can see so well under so many different conditions effortlessly is something of a miracle, because the brain does an amazing information processing job. You can think of the brain as basically a computing device that tells us a view of the external world. And more importantly, that information is not available to the brain unless these photons are captured by the retina of the eye. And under some conditions, the retina, the human eye, is probably the most sensitive photodetector of its kind. So I'm going to talk about detection of single photons by biological systems, what can offer us in terms of knowledge for other detectors and for other computing devices, and then talk about more recent work I've been doing on Fock states, which are a quantum optics state, which are just states containing just pure photons, and how we can measure them, which can lead us to better quantum computing devices, because you can detect or hold individual few quanta as such. Now, for physicists, the visual system is a good model, because you can study both wave and particle-like behavior of light. And all of you know from your elementary biology that there are about 100 million there are two classes of photoreceptors, rods and cones in the human eye, and there are about 100 million rod photoreceptors and about 25 million cone photoreceptors. Now, I'm going to talk primarily about rods, but a lot of what I say is applicable to cones also. Now, each rod is capable of detecting the absorption of a single photon. You don't have to cool down your detector or anything. And you can detect these at a level set by noise in the system. Now, the absorption probability of photons is approximately half, and detectable output signals with an efficiency of about 0.6. And the other amazing thing is that you can detect light over an enormous range. The operating range is huge, from starlight to bright sunlight, about 10 log unit range of photon fluxes. So the problem is that good rod vision requires very efficient photon counting, because a large fraction of photons are incident on the cell. They must be absorbed. And the absorbed photon must more often than not activate the molecule absorbing it. Two, the energy of the absorbed photon must be amplified so that it can produce a sizable electric current. The dark noise should be low, and the elementary response should have constant shape and size so that one photon can be distinguished from 0 and 2 photons reliably, or more. So actually, it was Lorentz, whom we all know, the Lorentz transform. It was Lorentz who actually first talked about how many photons, how many quanta do you need in order to just detect a flash of light. And Lorentz in 1911 actually did some calculations and showed that a just detectable flash delivered fewer than 100 quanta to the front of the eye can be detected. Of course, you have to take into account the scattering of light and the absorption of the media and so forth. So before I go into more greater details, I'll give you a quick elementary tour of the physiology of the eye. Now, this is a standard analogy that is given, that the eye is like a camera, which is not. Is there a pointer? Yeah. Oh, there it is. That's what it's looking for. Okay. The eye is like a camera, and I do not like this analogy, except for maybe the front part of the eye in terms of the dioptrics, in terms of image formation, which can be described very well by geometric optics. But once the light falls on the retina, it gets far more complex, because there are all types of nonlinear interactions that occur, and a lot of processing takes place at the level of the retina itself. Now, this is called the fundus. This is the back. If I were to take a high-powered lens and look into your eye, what I will really see is this region called the macula, which is the region of very fine details. This is very rich in cones, which operate under bright light conditions, what are called photopic conditions, and are responsible for your being able to see different colors. And out in the periphery, you have more rods. And the optic nerve is your so-called blind spot. When you learn to drive, people tell you to watch out for your blind spot, and that's what the optic nerve is. And the reason is, it's a region where there are no photoreceptors, so you cannot see them. So the organization of the retina is funny. First of all, there are a number of different retinal layers, but more importantly, you would think that here is a photoreceptor layer. Okay, this is where the rods and cones are. You would think that the photoreceptor layer would be at the front of the retina. Light is coming in this direction. But in reality, what happens is, there's a whole bunch of retinal layers. There's the outer nuclear layer, the ganglion cells, the inner plexiform layer, inner nuclear layer, and so on, all the way up to where the photoreceptors are. One of the reasons that it's organized in such a way is there's going to be a lot of light scatter. And this type of an arrangement helps to capture these scattered photons. Okay? I'll talk a little bit more about it later on. So here is a photoreceptor layer that's of interest to us. Like I said, there are two types of photoreceptors, rods and cones. The cone, as the name implies, do look like cones. And here you can see the rods, and here are the cones. Now, the important thing is, light comes in through here, is then propagated all the way to what is called the outer segment, which is at the way at the back. The outer segment is of interest, and the other reason it's of interest is because the way the light is captured and transmitted to sites of absorption out here, the photoreceptor behaves like a classical fiber optic element. And in fact, you can measure waveguide modal patterns in photoreceptors. In fact, that's how I got into doing vision research. When I was at Berkeley, I measured some of these waveguide modes in photoreceptors. So then, if you look into the outer segment, there is still further structuring. There are these, what are called discs. There are lots and lots of discs in there. And each disc is a lipid bimembrane. And if you look at the disc, each disc looks like this. You have these, you have this lipid bimembrane. Here are the discs, and embedded in it is your opsin molecules, rhodopsin in rod cells. And essentially, what it is This is a big protein. But in the protein, somewhere in there, is a chromophore that captures the photon. It's called retinal. It's an aldehyde of vitamin A. And essentially what happens is, here is your retinal, and it is in a form, it has a certain geometry. When it captures a photon, goes into an excited state, undergoes a whole series of changes in its conformation. Okay, and when it does that, it changes this rhodopsin structure, and that then gives rise to certain changes in the cellular membrane, which changes the transmembrane potential. Now, the other interesting thing, which actually helps to minimize stray-like noise, is that the eye is directionally sensitive. What that means is, if the eye were like a photodetector, it would not matter in which direction light came from. It would give the same constant output. But in reality, what you find is you are more sensitive to light coming in through the center of the exit pupil of the eye than to light coming from the periphery. And this is directly due to the waveguiding effects of the photoreceptors. It also means that photoreceptors are directionally sensitive, that you are sensitive to light coming from the periphery rather than from, less sensitive coming from the periphery than the center. It also means that photoreceptors are oriented like so. So they are not all, perpendicular to the underlying pigment epithelium, but there's a differential orientation which further helps to minimize stray-like noise. And one of the reasons that nature evolved waveguide properties is because that way your effective capture area for the photon is greater than the geometric size of the photoreceptor. Now, this is from an old article of mine, and this is how one models photoreceptors as a waveguide. Here, the direction of incident illumination is through here. You have the myoid and the inner segment. There's this is the ellipsoid area where it kind of tapers, and then here's a rod outer segment, and you can measure or calculate, make guesses about the permittivities of the various materials and the refractive indices. And in fact, these are some actual waveguide model patterns, which were measured from human photoreceptors, and the theoretical, this is the fundamental mode, TE00, TE01 mode, and so on, and these are the theoretical equivalents which you get from solving Maxwell's equations. Okay? And in fact, I like this figure because this is a visualization of the electric field values by solving the vector Maxwell equations. This was done on a supercomputer by Pickett et al. At Northwestern, which actually shows you the various wave patterns for different wavelengths. Okay, now we are going to talk about where vision begins. We are going to talk about rod photoreceptors. So one of the reasons theorists like me like to work with rods is because the geometry is simple. Now, this is a rod photoreceptor of an amphibian, essentially, here is your outer segment that I showed you earlier. It's packed with approximately 1 billion molecules of rhodopsin. This is the typical size, about 25 microns. And here's your inner segment, which has the mitochondria, which gives the energy to the cell, and so on. And basically what happens is when light is captured in the outer segment membrane out here, the ion channels close in response to light. There's a change, there's a hyperpolarization, there's a change in the membrane current. And in the outer segment, the current is shaped into a voltage signal. It connects by a synaptic junction to other cells. So the voltage signal then causes a neurotransmitter release that goes to the other end of the synapse and causes a bunch of other changes. And this is the rhodopsin molecule. It's an organic pigment. Retinal, which is covalently bound by the protein opsin. Here's your 11-cis retinal. You can measure its absorption curve and so on. So absorbing a photon triggers 11-cis to all trans isomerization. So it goes from this bent shape to this. And the protein responds to isomerization and structural changes. That's start a cascade of biochemical events. There are some interesting things about this. Because opsin acts like an electronic state selective catalyst. Now, if you're looking at, here are the time scales, okay? If a photon triggered isomerization, for retinal alone, it's about 1 nanosecond. The cis to trans isomerization takes place in about 1 nanosecond. For rhodopsin, the whole change takes about 200 femtoseconds. But if it were thermally activated, that is spontaneous isomerization, for retinal alone, it's about a year. For rhodopsin, it's about 1,000 years. Now, if it were about the order of a nanosecond, it's slow, so fluorescence can be competition. 200 femtoseconds, it's so fast that it competes with laws of quantum coherence, and you cannot go faster than laws of coherence. You know, your Schrodinger cat EPR paradox. Now, the thermal isomerization is a minimum dark noise level, and I can tell you that thermal isomerization takes place all the time. Consider that each rhodopsin, each rod outer segment contains over 100 billion rhodopsin molecules, okay? And there are 100 million photoreceptors in each eye. You can see this dark noise. Go sit yourself in a really dark room, dark it out for about half an hour, and you'll occasionally see points of light. That is rhodopsin spontaneously undergoing changes, isomerization. Or another way to visualize this is press on your eye. You'll see light. That's mechanically induced isomerization. So the thermal isomerization is the minimum dark noise level in the system, and in approximately 1 year to 1,000 years in retinal and rhodopsin, what happens is it reduces dark noise by more than 1,000 times. So everything in the system is so well optimized that behavioral noise level is approximately of the order of thermal isomerization. Now, on the other hand, if rhodopsin had the same properties as retinal, then you would not be able to see at light because there's a lot of noise would be coming in which would interfere with your ability to see. Unfortunately, we don't understand all of the implications of these numbers. Okay. This looks complicated, but it's really simple. Chemists have done a lot of work on this, and biophysicists have modeled this in terms of a series of coupled differential equations. Basically, what happens is, in the dark, there is a constant flow of ions. Sodium goes in extracellularly into in here, and potassium goes out. I can stick an electrode in and measure it. But when light hits rhodopsin, what happens is it undergoes a conformational change, this disc membrane. It induces something called a transducin, which activates an enzyme, which takes cyclic GMP and turns it into 5' GNP, which then blocks the flow of sodium into the membrane, and therefore you get a hyperpolarization wave. Now you can do some basic, assuming that the photon level is Poisson distributed and so on. You can show that that with conventional light sources, actual number of photons that arrive by the Poisson statistics. And so we can see when at least k photons arrive, then you can determine a probability of seeing and show that it is proportional to alpha times I. So that it's going to be proportional to the intensity. So you can measure the threshold K by looking at the shape of the curve. That is, if you plot the probability of seeing versus logarithm of I, then you can find the photon, you can measure what K is going to be. And these are called frequency of seeing curves. And what I plot here is the light intensity in arbitrary units and the probability of seeing for 3 different values of K. Okay? This is calculated from the previous equation, and depending upon what your threshold is, there are different shapes. Now, the classic work on this was done in 1942. It is such a beautifully done experiment. I tell my grad students to read the original paper because it is so well thought out and the analysis was so well done by Hecht, Schlaer and Pirenne at Columbia University in New York. It appeared in Journal of General Physiology. They were physicists in the physics department at Columbia, and they wrote a paper called Energy, Quanta, and Vision. And this was their old-fashioned optical system to calculate how much energy you need for vision. And everything was, without going to great details, I can tell you about it later, or even better yet, I would advise you to read this article. What they basically did was the number of observers, well dark adapted, measured in rod-rich area, light of wavelength which is maximally absorbed, and so on. And basically, what they plotted are frequency of seeing curves for 3 observers, Selig, Hecht, and maybe Stevens, and so on. But here they show each point represents 50 flashes, and here they are the logarithm of the average number of quantal energy versus flashes seen, and you get these frequency of seeing curves. And essentially what they did was they then compared these curves to curves generated assuming a Poisson statistic for the light, and basically they determined what K should be for a 50% or 60% probability of seeing. Now, independently, Van der Velden in Netherlands published in Physica similar studies which also measured frequency of seeing curves and the minimum number of quanta necessary to see for various values of K, again 1, 2, 3, and 4. And what the classic experiments really say is that for a dark adapted visual system, you can detect absorptions of 5 to 7 photons. 5 to 7 photons are all that you need in order to psychophysically press a button to say that you see a flash of light. Now, the way Hecht, Schlaer, and Pirenne did their experiment where device was that photons were incident on an area containing 500 rods. So you can do an analysis and show that individual rods must have detected single photon absorptions. Not a single rod would not have absorbed 5 to 7 photons, but 5 to 7 rods would have absorbed 1 photon each. Yeah?
But these were done without human subjects?
No, this is human subjects. Yeah, that's why I'm doing a frequency of seeing curve with a 60% probability. Later I'll show you single cell measurements that I've done.
The person puts a button and says, yes, I do.
Yeah, and you do repeat them. There are a number of statistical techniques to deal with this, and then we'll talk a little more. That's a very good question, but I'll show you. At that time, the technology wasn't there to do single unit work, but I'll show you some of that. Now, Van der Velden, on the other hand, showed essentially that you needed 2 absorbed photons for threshold. And he had higher false positive results, so thresholds were higher. Now, Barlow, actually in the Journal of the Optical Society of America in 1956, was the first to talk about intrinsic noise in the photoreceptor system. He called it as dark light, and he said it was indistinguishable from real photon absorptions. And Barbara Sackett, who was a grad student at Berkeley, actually wrote a very funny, interestingly titled paper called Counting Every Quantum, and was able to show that the visual system literally counted photon absorptions with a reliability limited by occasional photon-like events. What do I mean by photon-like events? Dark light. Also, and she also showed that rods produce signals that will allow neurons to discriminate between 1, 2, and 3 photon absorptions. So the moral of the story is individual rods can detect single photons. And individual rods can detect not only single photons, but they can trigger this whole biochemical cascade and neural events that come after it and processing to tell the brain that I have seen a photon. And for an observer to psychophysically respond at 60% probability that I have seen a flash of light, you only needed anywhere from 3 to 7 photons to be absorbed in order to say that I see a flash of light. Okay, here are recordings from single rods. This is some work I did as a grad student. And essentially what I have is I have a pipette, a suction pipette, and into which I draw a live cone or rod cell. That's my cell. I can give a flash of light, which is shown here, at at particular wavelength, change the intensity, so I can change the photon number. And this is my schematic, the equivalent electrical circuit. And this is now this figure shows the mean current in response to lights of various intensities. Now the current is of the order of a picoamp. And so the smallest response here is due to 4 photons, successive flashes are brighter by factors of 4. And here's the current responses to repeated dim flashes at times indicated by tick marks. And this is your current that you're getting, and this is about 1 picoamp. So now you cannot tell. Now, hidden in this is your dark noise, or photon-like events, which is the retinal spontaneously isomerizing. So, there are some real issues to be considered. Now, once a photon triggers a structural change, the cell needs to detect a single molecular event and amplify to a macroscopic level. So the macroscopic output is the current from closing ion channels in the cell membrane. But it is already a problem because the single photon current is about 1 picoamp, which is so close to the typical current through just one open channel in one rod cell. There are literally millions of channels in each cell. So, but we don't see the single channel noise at all. Now, channel noise is suppressed by having the light channels flicker rapidly between closed and open states. How it is done, we are not sure of the mechanism. So the fixed and inescapable noise variance is spread over huge bandwidth, which lowers the density in the relevant range, because the range is approximately of the order of 1 hertz, but the bandwidth is over 1,000 hertz. So the output actually is a change in probability of being open for many channels. So there are many such design features in the biochemical amplifier. Maybe it is something that we might borrow when we try to design amplifiers using nanotubes and so on. There's lots of examples from biology that have been used. And there are also many universal problems of photon counting. Okay, let's come to dark noise. Here's your measurements, again from the same type of system I showed earlier. This is the light. In the dark, I see these peaks. These peaks are your dark noise, spontaneously occurring. I'm not sending any light into my micropipette, just measuring continuously. And I can do some analysis, like for example, I can Fourier it and show what the what the current is per hertz and wavelength along hertz and so on. And here is my typical dark noise, which is about the same as the signal sent by capture of a photon. Baylor at Stanford was the first to show that you can detect single photon responses from toad photoreceptors, and single photoreceptors in the dark show photon-like spontaneous events at the right rate to be the source of dark noise. So Barlow identified a maximum dark noise level. If dark noise were anything above that, you will not be able to detect. So the conclusion is real rod cells have essentially this level of dark noise. Therefore, the visual system is operating at a level of reliability set by thermal noise alone, which is incredible sensitivity. So in the lab, you can lower the noise level even further by cooling the system. And in fact, Hecht, Caltech, what he did was he lowered the retinal noise with low body temperature, allowing for very high visual sensitivity, and trained them on a behavioral task and showed the threshold for reliable striking as a function of temperature. All these shows that there's a good match between behavioral and cellular noise levels versus temperature is perfect. Visual processing of dim lights is limited by input noise and not by any inefficiencies in the processing or by neurobiology. Hope I'm doing for time. Now there are, now let's say that you're doing a behavioral task. We don't want to talk about toads and other amphibians and so on. What about humans? Now people adjust the threshold for detecting signals in response to different situations. So all sensory tasks involve discrimination between signal and noise. Therefore, different strategies are used, and you end up with different types of errors. And this is the domain of signal detection theory and what is called the ROC analysis, which many of you might be familiar with, receiver operating characteristic. In fact, the application of signal detection theory, statistical signal detection theory to sensory systems was pioneered at Ann Arbor, the University of Michigan, actually, in the electrical engineering department. And essentially, what the human being or the brain does computationally is what we do in the physics lab. You detecting a signal is a problem of discriminating against noise. Now, you have also used, many people have used, like for example, Melvin Teich at Boston have used quantum optics techniques, for example, to manipulate photon arrival times with the retina using photon bunching and anti-bunching, name and type A distributions, and show that these trade-offs that observers make in terms of detecting signals from noises for different types of errors are changed with the expected results. This is a good paper in biological cybernetics. Now, you can have two types of noise, both additive and multiplicative noise, and this basically shows you for various frequency of seeing curves and the threshold number of photons that you need, and I won't go into great detail about it. There's some other interesting stuff I want to show you about. Okay, there are two components of dark noise. Now, this is from a primate. You can have a continuous noise as well as discrete dark noise, and essentially what you're seeing is, here is the response from about, I think, 8 discrete noise events with the cell's average single photon response. And but the amazing thing is, even though the dark noise is at the level of the signal, you can still differentiate between them. Now, here's a bit of modeling. So, of course, this is a little bit of hyperbole that you can measure everything. So, rod cells produce a current. Here's my current I as a function of time, which I write as some I naught to response to single photons at time t sub I plus some noise. So, I give some continuous background noise. And what does the brain want to do with these currents? So noise is obviously irrelevant, should be suppressed. And even more, and another additional factor is photon arrival times are not relevant by themselves for a very simple reason. The visual system does temporal summation. It summates these photon arrivals over a period of 100 milliseconds or so, depending upon the conditions under which you're measuring. So because of this temporal and spatial summation and so on, and the statistics of photon arrivals under normal conditions is Poisson, only things of possible relevance to behavior functions of the photon rate. So if you're doing modeling, it's tempting to write it in terms of a statistical inference problem. That is, rod current is related directly to photon arrival rate. But this is not right, because presumably the brain isn't interested in the arrival rate, but which is the light intensity, but in some features. So what type of features do you want? So at low light intensities, it doesn't matter because you have sufficient statistics. You can take the probability of arrival rate and the intensity is only a filtered version of the rod current filter, which could be a matched filter against spectrum n of omega. I can do this. This is from a very nice paper by Bill Bielek at Princeton. And you can show that, again, you can measure these and measure what the match filter should be and show that this filter should be implemented at the first stage of visual processing. Now, again, if you do that, you're going to the next level of the cell, which is the bipolar response. There's the measured bipolar response, and this is the time after flash, and here's the rod voltage response, and it seems to fit very nicely. But these predictions out here come from measured rod signals and noise and not the synaptic mechanisms, which we don't know very much about yet. And then what happens is the other thing. This is for one rod cell. Now we've got millions and millions of rod cells. So signal to noise for one photon in one cell is approximately 10, but if you look at one photon in greater than 100 cells, it appears as though you need some kind of nonlinearity in there. And field has shown that this is so, that there seems to be a nonlinear signal transfer that has implications for visual sensitivity. Okay, and there's lots more. And some of these give rise to, for example, illusions like something called the pull-frick effect, but let's not go into that. There is also another interesting thing that the visual detector does, which is the principle of univariance. We talk about wavelength, but once a photon is absorbed, all information about wavelength is lost. It doesn't matter what the wavelength is. So this basically shows the quantum response to different wavelengths, 460, 580, and 700 nanometers, and reproducibility of single photon responses. So, okay. This is the histogram is the responses due to 410 dim flashes, 129 responses, okay, and the time-dependent variance of the responses is shown here. I can measure all of this. Okay. So how does the photon, how does the rod compare to commercially available detectors, photomultiplier tubes, in terms of dark noise or CCDs and so on? This is, it gives you the quantum efficiencies of rods and cones, 0.25, 0.05, not as good as the CCD camera. Dark noise is much lower. Integration time of the order of about 300 milliseconds, 50 milliseconds for cones, 100 milliseconds for CCDs, and less than 10 to the minus 5 for PMTs. There are the typical pixel areas. Okay, so my summary is that at low light levels, visual system detects and counts photon absorptions with a reliability close to limits set by statistical fluctuations in the number of absorbed photons. There are many unanswered questions. What mechanisms are responsible for reproducibility? How are they reliably transmitted to the rest of the visual system? Molecular basis for differences in kinetic sensitivity and dynamic range. And more general questions. We can study this at many different levels, from level of single molecules. In fact, I used some quantum mechanical programs to actually look at the process by which retinal undergoes its changes, its vibrational states in the phase transition for pi-electronic states. And so you can look at it at the level of single molecules, to the dynamics of rhodopsin, single level of single cells. How do you get biochemical reaction cascade of individual molecular events to macroscopic transmembrane currents? Level of the retina, what are the rules by which these signals are integrated and encoded into the stereotype pulses that are the universal language of the brain, and the level of the organism. How does the brain make the discriminations required for optimal performance? Okay, so far I've told you about all of this, basic ideas on single photon. Now I'm going to talk about some new work on generating single photons using entanglement, and how Bob Boyd, though he's about a decade older than I am, is a lot more active. He was a senior to me at Berkeley, got his PhD with Charles Towns, and he's now at the Institute of Optics at Rochester. Okay, I'll try. So we're going to talk about, yeah. No, we do not have, humans, mammals do not have polarization sensitivity. Some bees do, certain fishes do, but I don't know what the evolutionary importance of that is.
So there are two different kinds of rods.
There are different types of rods, but we just don't seem to have a polarization sensitivity. Okay? Though the cornea is birefringent and so on, but we are not sensitive to polarized light.
How do these sensitivities compare to those in photosynthesis?
Humans are much better compared to photosynthetic mechanisms. Okay. The closest I can talk about is something called bacteriorhodopsin. Bacteriorhodopsin was found it's a bacteria in very high density salt marshes in the San Francisco Bay Area. And they have a similar chromophore. And rather than going through this neural process of a transmembrane current and all that, bacteriorhodopsin, they absorb photons and use a proton pump and directly convert that into energy. They have the closest efficiency to human rods. So for those who don't know what a Fock state is, a Fock state contains a definite number of photons and displays no intensity fluctuation. And in fact, what Bob and I are planning on doing is generate Fock states where we can measure the total number of photons and get better statistics on photon detection by the visual system. But the problem in generating Fock states is it can be experimentally very challenging to produce light fields that contain definite numbers of photons. Now, one of the simplest ways to measure generating single photon states is using spontaneous parametric down conversion, and this was first suggested by Hong and Mandel. What they did is they used a single photon detection of one photon of the pair to herald the presence of the other photon of the pair. And this is generalizable to multi-photon states of light. So if you have an unseeded optical parametric amplifier, OPA, pump photons, they interact nonlinearly with a non-centrosymmetric crystal, produce multiple pairs of less energetic photons. This is the term the signal and the idler through the process of down conversion. So the number of photons found in the signal and the idler fields is random, and it obeys thermal statistics, and it can be characterized by the gain of the OPA. Since every signal photon requires simultaneous emission of an idler photon, if one knows the number of photons in the idler field, then the number of signal field is determined with complete certainty. Therefore, by allowing transmission of the signal field only when the idler field is found to contain n photons, the signal field will be guaranteed to also contain exactly n photons. And Holmes et al. Were the first to talk about this for ideal photon number resolving detection. But if you want to do this, you need to create heralded multiphoton states, and it's not trivial to implement, and there's a lot of work being done in this. And a lot of what I'm going to talk about in the rest of this talk, where I have 10 minutes, is to give a possible scheme for doing this, for generating these spark states. And some of this has been published this year in Physical Review A, and we have some more work coming in. So the source of the photon pairs is the output of an unseeded OPA, and the heralding detector is a time multiplex detector. So what happens is the TMD provides photon number resolution by first randomly splitting the incoming state into many pathways that are separated in time from one another. That is, you put them into time bands, and the number of photons in the incoming field can be estimated to be equal to the total number of photons detected by two avalanche photodiodes. So this is basically the idea. You have an OPA, you pump it, the signal is a heralded field, you have an idler, so the heralding signal is sent through this loop, and at every point I have M50/50 splitters, and they go through and come to two avalanche photodiodes, and then I look at the output. So basically what I do in this calculation is I take a Bayesian approach, and for simplicity I assume that a system has 5 beam splitters, and here are the number of incident photons, uppercase N. I define a quantity called M, which is 2 raised to the power of M, so there are 32 different particle states, number of detected photons, and I give, so the colored portions are the significant probability of detecting N photons conditioned on uppercase N photons being incident on the multiplex detector. And what I'm doing is I'm giving different values for single photon detection efficiencies, 1 to 0.33. So there are a number of counting errors, and so if I have a system containing M beam splitters, I will have M uppercase M, oops, that's an error, 2 raised to the power of M different time bins. So if the number of photons N is much smaller than M, you could have undercounting due to imperfect splitting. It's going to be small. But on the other hand, if N is comparable to M, you must account for splitting errors. So use a standard EARN model. You can calculate the conditional probability of detecting N photons given that the incoming field has uppercase N photons in it. And you get something of this, which depends upon the eta, the efficiency of your photodetector, and okay. And I can do a Bayesian analysis and show that the probability of the gain. So the photon number distribution for either the signal or the idler field will obey Bose-Einstein statistics, and is given by an expression of this form. And I can show you can get the resultant mixed state of the signal field is given in terms of, given that the answer by photons are detected in the idler field. I can write a density matrix and then go through and apply Bayes' theorem to get an estimate and estimate the number of photons in the signal field using a maximum likelihood estimate. And if I do that, this is what I get. For example, again, if I assume a Poisson distributed field with a mean number of photons as being 5, here's your photon field, and this is the heralded field with what's the probability if there are 4 excuse me, for a typical heralded field. And so this is conditioned on the detection of 4 photons in the idler field. And anyway, I can go through all of this, and and then look at the mean squared error and so forth. I can get what the uncertainty in the number of photons in a heralded field when N sub I photons are detected in the heralding field for different etas. And I can also use something called Mandel's Q parameter, which I plot here where there are so here I do that. If Q parameter is greater than 1, the light is said to be super Poissonian. Here is it's not Poissonian at all. And Mandel's Q parameter versus gain for the OPA and single photon detection efficiency for the TMD in the case where N sub ML is 5. Heralded field is a single mode estimated to contain 5 photons. So I can now go through this and do contour lines of constant probability of creating a Herald state. And here I'm assuming there are 5 50-50 beam splitters. I can expand this analysis to look at multimoded states, and if I do that, the probability that the OPA will emit k pairs of photons distributed among mu modes is given by something like this, and I can do a similar analysis. So having said all of this, I will leave this with this quote from Einstein. When we talk about quantum systems and entanglement and all that, it's nice to know what a photon is, but I kind of like this. This was Einstein writing to his pal Bessel in 1951, and I don't think we know that much more about the photon since then either. So I'll stop at this. Probably next year I'll be able to show, using the Fox state generation of photons, much better results in terms of much better statistics in terms of photon detection in live humans rather than single unit measurements. This probably has other implications for quantum computing, this type of a scheme to detect single photons. Let's stop at this. Thank you.